I am Dr. S. Karpagam, Associate Professor of Botany, Queen Mary's College, Chennai, India. I am presenting the paper Preliminary Phytochemical Screening and Antimicrobial Activity of Cotyledon of two varieties of Mangifera indica. The authors are Arul, Pamila and Karpagam. India is the largest producer of mangoes. There are hundreds of varieties of mangoes in India, each with a distinctive taste, shape and size. Worldwide, hundreds of mango cultivars are grown. In India alone, there are around 280 types of mangoes, out of which only 30 are well known. Mango, the most important fruit of India, is grown in an area of 1.23 million hectares with an annual production of 20 million tons, which accounts for 57% of the total world production. Mangifera indica belongs to the Anacardiaceae family, is a large evergreen tree of tropical and subtropical regions of the world. It is one of the choicest fruit, especially in Asia. Mango is popular due to its excellent flavor, delicious taste, delicate fragrance, attractive color and nutritive value, which make it rank among the best fruits of the world. Though it is a very large tree with a spreading canopy, nowadays the tree is grown as a smaller tree due to the grafting experiments. The tree flowers in the early months of January and it starts ripening in the months of April and the season extends up to July. In the present study, two varieties, namely the Banganapalli and Rumani, are taken. The Banganapalli is an early producer which starts ripening in the months of April, and it is a large fruit with 300 to 400 grams per fruit. It has grown all over India, but it is most famous in the village Banganapalli in Andhra Pradesh, and it has received a geographical indication tag in 2017. The pulp is fiberless, firm and yellow with a sweet taste. The Rumani is other tasteful fruit, but it's grown in the southern India and it is grown commercially for its light yellow colored fiberless pulp and a sweet rich taste. It is a late bloomer. It ripens in the late season in the months of July. All the parts of the plants are rich in tannins and flavonoids. The tree is a good source of timber. The different parts of mango have a broad range of medicinal properties such as antimicrobial, antiviral, antifungal and anti-inflammatory properties. It has anti-diarrheal and antioxidant effect. It is a very good immunomodulatory effect. The nutritive value per 100 gram of mango pulp is very high, that it contains 14 gram of sugar. The carbohydrate and dietary fiber is also very rich. It contains calcium, iron and magnesium, and it is a rich source of vitamins and minerals. Each serving of mango is fat-free, sodium-free and cholesterol-free. The mangoes contain over 20 different vitamins and minerals, helping to make them a superfood. 1 cup of mango provides 50% of the daily need of vitamin C, 8% of the daily need of vitamin A and vitamin B6. The world production of mango is about 40 million tons. India is the largest producer with 20 million tons annually. 100 gram of mango seed kernel has more nutritional value than 500 gram of potatoes and 2 kg of mango pulp. Mango kernel has 20-fold higher protein, 50-fold higher fat and 4-fold higher carbohydrate than mango pulp. The kernel has balanced quantities of protein, carbohydrate, oil, vitamin, mineral and therapeutic phytochemicals in sufficient quantity. Yet we can eat the mango pulp, which has only less nutritional value, and we throw away the kernel, which has almost 20 to 50 times more nutritional value than the pulp. When we thrash the kernels, not only we are polluting our land, but we are throwing away a highly nutritious food. The objective of this article is to make people aware of the nutritional value of the mango kernel and promotes its utilization. The Banganapalli seeds are large with a monospermic cotyledon and large cotyledons. The Rumani seeds are comparatively smaller when compared to the Banganapalli, and the cotyledons are also smaller. The health benefits of mango cotyledon is immense. It is hair care. It can be used to control dandruff, prevents hair loss and it improves the hair health. It is also a very good cardiovascular disease. It improves the female reproductive health. It also improves digestion, controls diarrhea. It is used in the dental health, especially it is used as a throat gargle. It is active against diabetes, obesity and cholesterol, and it also protects the skin against acne. And it is a very good for anemic. The nutritive content of mango kernel flour is high, that it contains carbohydrates, proteins and fat in abundance, and it contains the minerals calcium, magnesium, sodium, potassium and iron in large quantities. That's why it's very effective against anemia treatment. The quality of the mango seed kernel is as a high specific gravity, refractive index, iodine value and saponification value. In the present study, two varieties of mango, the Banganapalli and the Rumani seed kernels were collected, air-dried, powdered. 10 grams of the powder was soaked for 24 hours in 50 ml of the organic solvents, namely ethanol, petroleum ether, acetone, chloroform and additionally aqueous. They were filtered, dried and redissolved in the respective solvents. The phytochemical screening was carried out by using the standard protocol as described by Harbo 1973. Alkaloids, carbohydrates, saponin, phenol, flavonoids, diterpenes, tannins, terpenoids, protein, steroid, coumarin, quinine and xanthoproteins were estimated. The antibacterial activity was evaluated by well diffusion method using Miller-Hinton agar. The selected microorganisms were Staphylococcus aureus, Enterococcus faecalis, Klebsiella pneumonia and Escherichia coli. Amoxicillin was used as the reference. The antifungal activity was tested in the potato dextrose agar medium. The fungal cultures used were Candida albicans, Epidermophyton floccosum, Aspergillus niger. Ketoconazole was used as the reference. The phytochemical content of the two varieties of Mangifera indica, namely the Banganapalli and the Rumani seeds, shows that the activity is more in the aqueous extract when compared to the solvents. The ethanol also has a higher phytochemical content. Of the two varieties, the Banganapalli shows more phytochemical in both aqueous and ethanol when compared to the Rumani variety. The most abundant phytochemicals are saponins, phenols, flavonoids, diterpenes, the tannins. The antimicrobial activity of the acetone extract of Mangifera indica shows a moderate activity at a higher concentration of 50 micrograms. Whereas the aqueous extract has an antimicrobial activity even at a lower concentration of 30 micrograms. It could control the growth of Staphylococcus aureus and Candida albicans even at a lower concentration. Whereas the chloroform extract does not show much activity. The ethanol extract also shows a higher activity, but even at a lower concentration, it's able to suppress the growth of most of the pathogens. The petroleum ether does not show much activity when compared to the other solvents. Of all the byproducts of mango tree, its kernel is the cheapest and most readily available. Huge amounts of unutilized mango kernels can be used as feed additives to enhance the non-specific immunity. Mango seed kernel is one of the byproducts of food processing industry and is not commercially exploited but are discarded as waste. Since the kernels have a slight bitter taste, they are not commonly consumed. From the present study, the phytochemical content and antimicrobial activity of two types of mango seed kernel, namely Banganapalli and Rumani, were compared. But it was found that the Banganapalli is more potent antimicrobial agent than the Rumani seed. The investigation is underway in terms of toxicological studies and purification of active components from the view to use the mango seed in novel drug development. These are all the references. The author, Arul Pamila, did her post-graduation and doctorate from Queen Mary's College, Chennai, India. She secured the first rank in PG Botany. She published 12 papers in international journals, presented 9 papers in various national and international conferences, and she has won the first Best Paper Present Award from Meenakshi College, Chennai. The other author, Dr. Karpagam, is an associate professor and has 22 years of teaching experience and 26 years of research experience. She has published 43 research papers and guided 16 MPhil and 15 candidates. Any further queries or clarifications regarding this paper could be contacted through email. Thank you.